Close your eyes and listen. Not to the sounds around you, but to the silence within. In this moment, as you settle into comfort, something extraordinary is happening inside your body. Something so quiet, so gentle, that you have never truly heard it before. Your body is not still. It never has been. Right now, as you breathe slowly and deeply, trillions of tiny musicians are playing their songs inside you. Each cell in your body is like a single instrument in the most beautiful orchestra ever created. They hum together in perfect harmony, creating the symphony that is you. Think about this for a moment. Every cell in your fingertips, every cell in your heart, every cell behind your closed eyelids. They are all singing. They have been singing since the day you were born. They will continue to sing for as long as you live. What kind of songs do they sing? Your heart cells sing a steady, rhythmic beat. Lub-dub, lub-dub, lub-dub. Like drummers keeping time for the rest of the orchestra. They never miss a beat. They never take a break. Even now, as you rest, they continue their faithful rhythm. Your brain cells sing in quick, electric whispers. They pass messages back and forth like notes between friends. These messages travel faster than lightning, carrying your thoughts, your memories, your dreams. Right now, they are creating the very thoughts you are thinking about your own cells. Your liver cells sing a song of cleaning and healing. They work like gentle janitors, sweeping toxins from your blood. They transform the food you ate today into energy you can use tomorrow. Their song is one of service, quiet and constant. Your lung cells sing the song of breath. They welcome oxygen like old friends and release carbon dioxide like a gentle sigh. With every breath you take, they dance. In and out, matching the rhythm of your sleeping breath. But perhaps the most amazing singers of all are the cells that make up your skin. They sing the song of protection. Like a choir of guardians, they link arms and form an unbroken wall around your entire body. They keep the outside world outside and the inside world safe. All of these cellular musicians play together without a single conductor telling them what to do. How do they know when to start? How do they know what song to sing? How do they stay in perfect harmony? The answer lies in something even more amazing than the cells themselves. It lies in the ancient sheet music they all carry. This sheet music is written in a language older than any human language. It is written in the 4-letter alphabet of DNA. Inside every single cell in your body, called up like the tiniest scroll imaginable, is a complete copy of your genetic code. Think about this. Every cell carries the same musical score. The same instructions. The same ancient wisdom. But here is where the magic really happens. Even though every cell has the same sheet music, they each choose to play different parts of the song. Your heart cells read the heart section. Your brain cells read the brain section. Your skin cells read the skin section. It is like having a massive orchestra where every musician has the complete musical score, but each one plays only their own instrument's part. The violinist does not try to play the drums. The drummer does not try to play the flute. They trust in the harmony of the whole. Your cells trust in this same harmony. They have been doing this dance of cooperation for billions of years. Long before humans existed, cells learned how to work together. They learned how to sing together. They learned how to become something greater than themselves. As you lie here now, breathing slowly and peacefully, you are the living proof of this incredible collaboration. You are not just one thing. You are a community. You are a symphony. You are trillions of individual lives that have chosen to become one life together. Your skin cells will never see your heart cells. Your liver cells will never meet your brain cells. Yet they all work together with perfect trust. They share resources. They send chemical messages. They support each other in ways you cannot even imagine. When you are happy, your cells feel it. When you are peaceful, like now, your cells become peaceful too. When you laugh, the vibrations of your joy travel through every part of your cellular symphony. When you sleep, they adjust their songs to match your restful rhythm. You are not separate from your cells. You are your cells. And your cells are you. This is not poetry. This is the deepest truth of your existence. You are a walking, breathing, dreaming community of trillions of tiny lives. Tonight, as you drift toward sleep, you can rest in the knowledge that your cellular symphony will continue playing. They do not need your conscious mind to tell them what to do. They carry within themselves the wisdom of ages. Your heart will keep its steady beat. Your lungs will keep their gentle rhythm. Your brain will sort through the day's experiences and weave them into dreams. Your liver will continue its quiet work of healing. Your skin will stand guard through the night. This is the first miracle of your existence. Before you can even think about growing new organs or healing damaged tissue, you must first marvel at the symphony that already exists within you. But what happens when some of the musicians in your cellular orchestra need help? What happens when the ancient sheet music gets damaged, or when cells forget how to sing their proper songs? This is where our story begins to unfold, in the vast library where all of life's music is stored. Imagine stepping into the most beautiful library you have ever seen. The walls stretch impossibly high, lined with countless books that glow with a soft, warm light. The air smells of old paper and quiet wisdom. Your footsteps are silent on the polished floor. This library exists. It is real. And it lives inside every single cell in your body. Right now, as you rest in the comfort of this moment, trillions of these libraries surround you. They are smaller than the smallest grain of sand, yet each one contains more information than all the books ever written by human hands. Welcome to the library of your DNA. In this sacred space, there are no thick volumes or leather-bound books. Instead, there are delicate strands that spiral like the most elegant staircases. These spirals are your DNA, and they hold the complete instructions for building you. The language written in this library is surprisingly simple. It uses only 4 letters, A, T, G, and C are just for letters. Yet from these 4 simple symbols, the entire complexity of your existence is spelled out. Your brown eyes or blue eyes. Your laugh. The way your heart beats. The texture of your hair. All written in combinations of just 4 letters. Think about that for a moment. The English language uses 26 letters to write poetry and stories. Your genetic language uses only 4 letters to write the story of you. And what a story it is. These 4 letters do not exist alone. They pair up like dance partners who have been dancing together for billions of years. A always dances with T, G always dances with C. They hold hands across the spiral staircase of your DNA, creating the rungs of a ladder that twists gently through space. As you breathe slowly in and out, imagine these molecular dancers swaying in rhythm with your breath. They are patient dancers. They have been holding their position since the moment you were conceived. They will continue their dance for your entire life. But this is not a still, silent library. It is alive with quiet activity. Right now, in cells throughout your body, tiny librarians are carefully unrolling sections of your genetic scroll. They read the instructions written there with infinite care. They translate the 4-letter language into the proteins that build your body. Your heart cells are reading the section that tells them how to beat steadily. Your brain cells are reading the instructions for thinking and dreaming. Your skin cells are reading the chapters about protection and healing. Each cell type finds its own special section and follows those instructions with devoted precision. The truly miraculous thing is that every cell in your body contains the complete library. Every single cell. The cell in your big toe contains the same genetic information as the cell in your brain. Yet somehow, your toe cells never try to think, and your brain cells never try to grow toenails. How do they know which sections to read? The answer lies in the wisdom of the librarians themselves. These molecular librarians are proteins that act like gentle guides. They know exactly which books should be opened in which cells. They place tiny bookmarks on the sections that each cell needs to read. They keep all the other sections closed and quiet. In your liver cells, the librarians open the books about cleaning toxins and processing nutrients. They keep the books about making tears and growing hair tightly closed. In your eye cells, they open the chapters about seeing light and creating tears. They leave the liver books sealed and silent. This system of cellular librarians has been perfected over billions of years. Long before the first human walked the earth, cells learned how to read their genetic libraries correctly. They learned how to be exactly what they needed to be when they needed to be it. Your genetic library is not a new creation. The books in this library are ancient beyond imagination. Some of the instructions written in your DNA are the same instructions that have been passed down through millions of generations. The genetic code that tells your heart how to beat is nearly identical to the code that made your great-great-grandmother's heart beat. You carry within you the wisdom of ancestors you will never know. Fish that swam in ancient oceans. Small mammals that survived the age of dinosaurs. Early humans who first looked up at the stars and wondered. Their genetic stories are woven into yours, creating the tapestry of your existence. Yet your genetic library is also uniquely yours. No one else who has ever lived, or ever will live, has exactly the same collection of genetic books. You are a first edition. You are the only copy of yourself that will ever exist. The spiral staircases of your DNA stretch for incredible distances. If you could unwind all the DNA from just one of your cells and lay it out in a straight line, it would be about 6 feet long. All of this, called up in a space smaller than the period at the end of this sentence. Now multiply that by the trillions of cells in your body. The total length of DNA in your body would stretch from the Earth to the Sun and back again, several times over. You are a walking library of cosmic proportions. As you lie here tonight, your cellular librarians continue their quiet work. They read the instructions for repair and renewal. They follow the genetic recipes for healing small damages from the day. They prepare your body for tomorrow's adventures. Some of them are reading the sections about sleep, helping your brain sort through memories and dreams. Others are reading the chapters about growth, slowly replacing old cells with fresh new ones. Still others are reading the ancient texts about immunity, standing guard against anything that might harm you. This is the second miracle of your existence. Not only are you a symphony of cellular musicians, but each musician carries within them the complete sheet music for the entire symphony of life. But what happens when some of the pages in these ancient books become damaged? What occurs when the gentle librarians make mistakes in their reading? Sometimes, even in the most carefully tended libraries, accidents happen. Chapter 3, When nature makes mistakes. Sometimes the librarians of your cells grow tired, and copy errors slip into the sacred texts, like typos in a love letter that change its meaning. These gentle mistakes ripple through generations, creating the beautiful diversity of human hearts that beat differently. Even in the most perfect library, sometimes a page gets torn. Sometimes a word gets smudged. Sometimes a tired librarian copies a sentence incorrectly, and the mistake becomes part of the story forever. This happens in your genetic library too. As you rest here in the quiet of this moment, it is important to understand that your DNA is not perfect. No one's DNA is perfect. And surprisingly, this imperfection is not a flaw in the design of life. It is one of life's greatest gifts. Imagine a scribe in an ancient monastery, carefully copying a sacred text by candlelight. His hand is steady, his attention focused. But after hours of work, his eyes grow tired. His hand trembles slightly. A single letter changes. An 8 becomes a T. A G becomes a C. This is how mutations happen in your genetic code. They are usually small, simple changes. One letter swapped for another. One dance partner accidentally switching places with a different partner in the spiral staircase of your DNA. Most of the time, these tiny changes mean nothing at all. Your genetic library is so vast, so redundant, that a single typo rarely affects the larger story. It is like changing one word in a million-page encyclopedia. The overall meaning remains the same. But sometimes, rarely, a single letter change can rewrite an entire chapter of who you are. This is how you got your unique eye color. Somewhere in your family's genetic history, a tiny mistake in copying created the exact shade of brown or blue or green that you see when you look in the mirror. This mistake was actually a gift, passed down through generations until it reached you. This is how you got the particular curve of your smile, the exact texture of your hair, the way your laugh sounds different from everyone else's laugh. Each of these traits began as a small copying error in someone's genetic library long ago. Your cells are constantly making new copies of your genetic books. Every time a cell divides to create two new cells, the entire library must be copied perfectly. Think about this enormous task. 6 billion letters must be copied exactly, with no mistakes, every single time. The molecular librarians who do this copying work are incredibly skilled. They have been perfecting their craft for billions of years. They copy your genetic code with an accuracy that would make the most careful human scribe weep with envy. But they are not perfect. In all those billions of letters, occasionally one gets copied incorrectly. Sometimes the copying machinery skips a letter entirely. Sometimes it adds an extra letter that does not belong. When this happens during your lifetime, your body has remarkable ways of fixing most mistakes. Special librarians patrol the genetic books, looking for errors. They act like gentle editors, finding typos and correcting them before any harm is done. They are incredibly good at their job. But some mistakes slip through. And some mistakes happen in very important parts of the genetic code. When this occurs, cells may start reading their instructions incorrectly. A heart cell might forget how to beat properly. A liver cell might stop cleaning toxins effectively. A brain cell might struggle to send its messages clearly. These are not cruel punishments. They are simply the unavoidable consequence of being alive in a world where change is constant. Sometimes these copying mistakes happen in the very special cells that create new life. When genetic errors occur in these cells, they get passed on to children. This is how genetic conditions travel through families, like ancient stories told from parent to child. But here is the beautiful paradox of genetic mistakes. The same process that can cause problems is also the engine of all diversity and evolution. Without genetic mistakes, every human would be identical. Without these tiny copying errors, life would never change, never adapt, never become more complex or beautiful. Every difference between you and your siblings came from these gentle genetic mistakes. Every difference between humans and other animals arose from millions of years of small copying errors, building up slowly over time. The genetic mistake that gave some people the ability to digest milk as adults allowed entire civilizations to thrive. The copying error that changed skin color helped humans survive in different climates around the world. The small changes that created different blood types, different immune systems, different ways of processing medicines, all began as simple typos in the genetic code. So when we talk about genetic mistakes, we are really talking about the raw material of life's creativity. Each mistake is like a small experiment that nature runs to see what might be possible. Most experiments fail quietly, causing no harm. Some experiments cause difficulties that require care and attention. But some experiments create new forms of beauty, new capabilities, new ways of being human that never existed before. You are the result of millions of these experiments. Every cell in your body carries the history of countless genetic mistakes that slowly shaped your ancestors into the person who became your parent, and your parent's parent, stretching back through time to the very beginning of life itself. As you breathe slowly and deeply, you can appreciate that you are not just a perfect copy of an ancient genetic template. You are something new. You are the latest chapter in a story that has been writing itself for billions of years, one small mistake at a time. Tonight, as your cells continue their quiet work of copying and repairing your genetic libraries, some of them will make tiny mistakes. This is not something to fear. This is simply life being life, constantly creating new possibilities. But what if we could guide these changes? What if we could help nature make better mistakes, or fix the ones that cause suffering? Deep in laboratories around the world, scientists have discovered ancient tools that might make this possible. Deep beneath the ocean, in places where sunlight has never touched, tiny bacteria have been fighting invisible wars for billions of years. These microscopic warriors developed weapons so precise, so elegant, that they would eventually change the course of human medicine forever. You cannot see these bacteria with your naked eye. They are smaller than the smallest grain of sand, simpler than the simplest plant. Yet within their tiny bodies, they carry tools that can edit the book of life itself. This is the story of how scientists discovered these ancient molecular scissors, and how those scissors might one day help grow new organs from your own cells. The discovery began, as many great discoveries do, with curiosity about something very small. In the 1980s, scientists noticed something strange in the genetic libraries of bacteria. They found repeating patterns in the DNA. Short sequences that appeared over and over again, like stuttering words in an ancient text. At first, these patterns seemed like mistakes. Junk sequences that served no purpose. But nature rarely creates true junk. Everything in the genetic code exists for a reason, even if that reason is not immediately clear. Years passed. Different scientists in different laboratories studied these repeating patterns. Slowly, patiently, they began to understand what they were looking at. These were not mistakes. They were memories. The bacteria were keeping journals. In their genetic code, they recorded the signatures of every virus that had ever tried to attack them. Like an immune system with perfect memory, they collected pieces of their enemy's genetic code and stored them in their own DNA. But why would bacteria want to remember their enemies? The answer revealed one of nature's most elegant defense systems. These bacteria had learned to recognize returning threats. When a virus that had attacked before tried to invade again, the bacteria would consult their genetic journal. They would recognize the intruder's signature immediately. And then they would deploy their molecular scissors. These scissors are proteins, folded into shapes that allow them to cut DNA with incredible precision. They can locate a specific sequence among billions of genetic letters. They can find one particular sentence in a library containing millions of books. And when they find their target, they cut it out with surgical accuracy. Imagine having scissors so precise that you could cut a single word out of a newspaper without damaging any of the letters around it. Now imagine that the newspaper is smaller than the tip of a needle, and the word you need to cut is just 4 letters long. This is the level of precision that bacterial scissors achieve every day. The bacteria had been using these tools for defense. When they recognized an enemy virus, they would send out their molecular scissors to cut up the invader's genetic code. The virus would be destroyed, unable to function with its DNA in pieces. Scientists named this system CRISPR. The letters stand for technical words, but the meaning is simple. It is a way for bacteria to remember their enemies and cut them apart when they return. For years, CRISPR remained a fascinating curiosity. Scientists understood how it worked in bacteria, but they did not yet realize its true potential. It was like discovering a master key but not knowing what doors it could open. Then, in laboratories around the world, researchers began to wonder, what if we could use these bacterial scissors for our own purposes? What if we could guide them to cut specific parts of human DNA? The idea was both thrilling and terrifying. For the first time in history, humans might be able to edit their own genetic code with the same precision that bacteria use to fight viruses. We could potentially fix genetic mistakes. We could remove harmful sequences. We could even add new capabilities. But first, scientists had to learn how to control these ancient tools. They had to understand exactly how the molecular scissors recognized their targets. They had to figure out how to guide them to the right place in the vast library of human DNA. This required years of patient work. Scientists studied the shape of the scissors. They mapped every curve and angle of these protein tools. They learned how to modify the guide sequences that tell the scissors where to cut. Slowly, carefully, they learned to reprogram the bacterial weapons for peaceful purposes. They transformed tools of microscopic war into instruments of healing. The breakthrough came when researchers realized they could design their own guide sequences. Instead of letting bacteria choose what to cut, humans could write their own instructions. They could point the molecular scissors at any sequence they wanted to change. For the first time, the four-letter language of DNA became something humans could edit directly. We were no longer just readers of the genetic code. We could become writers. In laboratories that hummed quietly through the night, scientists began testing these reprogrammed scissors on human cells grown in glass dishes. They watched in amazement as the tools cut and pasted genetic sequences with elegant precision. They could remove a single faulty letter from a gene. They could replace it with the correct letter. They could cut out entire sections of harmful DNA and replace them with healthy sequences. The molecular scissors that had evolved to protect bacteria from viruses were now learning to protect human cells from genetic diseases. As you rest here tonight, breathing slowly and peacefully, CRISPR tools are being refined in laboratories around the world. Scientists are learning to make them even more precise, even gentler in their touch. But the most exciting possibility is still unfolding. What if these molecular scissors could be used not just to fix genetic mistakes, but to help cells remember how to grow into entirely new organs? Picture the most delicate artist you can imagine. Someone who can paint with a single hair from a brush, who can carve details so fine they are almost invisible to the naked eye. Someone whose touch is so gentle that they can repair a butterfly's wing without disturbing the dust that gives it color. This is how CRISPR works inside your cells. As you rest here in the quiet comfort of this moment, it is important to understand just how remarkably gentle these molecular tools can be. They are not crude hammers breaking down walls. They are not rough saws cutting through everything in their path. They are precision instruments that work with the delicacy of a master craftsman. When CRISPR enters a cell, it moves like a careful librarian through the vast genetic library we explored earlier. It does not rush. It does not force its way through the delicate spirals of DNA. Instead, it glides along the twisted ladder of your genetic code, reading each letter sequence with patient attention. The CRISPR system consists of two main parts working together in perfect harmony. The first part is called the guide RNA. Think of this as a molecular detective, carrying a photograph of exactly what it is looking for. This photograph is written in the same four-letter language as your DNA. The guide RNA moves through your genetic library like a careful searcher, comparing its photograph to every sentence it encounters. A, T, G, C. It reads each sequence, checking for the perfect match. Not almost perfect. Not close enough. Perfect. This search can take time. Your genetic library contains 3 billion letters, organized into countless sentences and paragraphs. Finding one specific sequence among all those letters is like finding one particular grain of sand on a vast beach. But the guide RNA is patient. It has been designed for this exact purpose. It will search for as long as it takes, moving gently through the coiled corridors of your DNA until it finds its perfect match. When the guide RNA finally locates its target sequence, something beautiful happens. It binds to those letters with a soft click, like a key finding its lock. The match is so precise that no other sequence in your entire genetic code will do. Only these specific letters, in this exact order, will satisfy the molecular detective. Now the second part of the CRISPR system awakens. This is the protein called Cas9, and it acts like the most precise scissors ever created. But these scissors remain completely inactive until the guide RNA gives them permission to cut. Think of Cas9 as a master surgeon who keeps their scalpel perfectly still until the exact moment when it is needed. The protein travels alongside the guide RNA, waiting patiently for the signal that says, "Here." This is the place. This is the moment to act. When that signal comes, Cas9 makes 2 perfect cuts in the DNA, one on each side of the target sequence. The cuts are so clean, so precise, that they create what scientists call the blunt ends. No ragged tears. No damage to the surrounding genetic text. Just 2 clean cuts that allow the unwanted sequence to be removed. But CRISPR does not simply destroy. It also creates. After the faulty sequence is removed, the cell's own repair mechanisms spring into action. These natural healing systems have been perfecting their craft for billions of years. They know exactly how to mend breaks in DNA. Scientists can guide this repair process by providing the correct genetic sequence to replace the faulty one. It is like giving the cell's repair crew a perfect patch to place over the damaged area. The new sequence integrates seamlessly into the genetic text, becoming part of the permanent library. The most remarkable thing about this entire process is how gentle it is. CRISPR makes its changes without disrupting the delicate balance of the cell. The cellular symphony continues playing. The molecular librarians continue their work. Life continues its ancient rhythms, now with a small but important correction to the genetic score. This gentleness is what makes CRISPR so different from earlier attempts to modify genetic code. Previous methods were like trying to edit a book by tearing out entire pages and gluing in new ones. The results were often messy, unpredictable, and sometimes harmful. CRISPR is like having a pen that can erase individual letters and write new ones in their place, without disturbing the ink around them. The precision is almost beyond human comprehension. Scientists have spent years learning to make CRISPR even gentler. They have discovered ways to make the molecular scissors cut more precisely. They have learned to design guide RNAs that are even more specific in their search. They have developed methods to ensure that the repair process happens exactly as intended. In laboratories around the world, researchers test each CRISPR system thousands of times before it is ever used in living cells. They verify that it cuts only where it is supposed to cut. They confirm that it makes no unwanted changes elsewhere in the genetic code. They ensure that the cell remains healthy and functional after the edit is complete. This level of care and precision has opened doors that scientists once thought would remain closed forever. Genetic diseases that seemed impossible to treat are now yielding to CRISPR's gentle touch. Cells that had lost their way are learning to function properly again. But perhaps the most exciting possibility lies not in fixing broken genes, but in awakening sleeping ones. Your body contains cells that remember how to become anything. They carry within them the potential to grow into hearts, lungs, kidneys, or any other organ you might need. These cells are like seeds that have forgotten how to sprout. They contain all the genetic information necessary for growth, but they need the right signals to awaken their dormant potential. CRISPR's gentle touch might be the key to helping these sleeping cells remember their dreams. By making precise changes to their genetic switches, scientists might be able to convince adult cells to become young again, to remember the unlimited potential they had when you were first forming in your mother's womb. As you breathe slowly and peacefully, imagine these molecular tools moving through cells in laboratories around the world. They work with infinite patience, making tiny adjustments that could lead to extraordinary possibilities. But before we can understand how to grow new organs, we must first explore the garden where these miracles begin, in the special cells that hold within themselves the blueprint for every part of your body. Deep inside your bones, in the soft, spongy center that you never see, lies one of nature's most extraordinary gardens. This is not a garden of flowers or trees. This is a garden of pure possibility, where special cells wait patiently to become whatever your body needs them to be. These are your stem cells, and they are unlike any other cells in your body. As you rest here peacefully, millions of these remarkable cells are nestled in the warm, protected spaces inside your bones. They live in a world of gentle currents, where nutrients flow like streams through a hidden valley. They are bathed in chemical signals that keep them young, keep them waiting, keep them ready. Imagine a seed that has never decided what plant it will become. In your garden at home, a tomato seed can only become a tomato plant. A rose seed can only grow into a rose bush. But stem cells are different. They are like magical seeds that could become any plant in the entire garden, depending on what the garden needs most. Your stem cells carry within them the same complete genetic library that every other cell in your body possesses. The same ancient books, the same four-letter alphabet, the same inherited wisdom from millions of generations. But unlike your specialized cells, stem cells keep all their books open. They read from every section of the genetic library at once. This is what makes them so special. A heart cell has learned to read only the heart chapters of your genetic code. A brain cell focuses only on the brain sections. A liver cell dedicates itself entirely to liver functions. But stem cells remain students of everything, masters of nothing, keeping all possibilities alive. When your body calls for help, stem cells listen. They receive chemical messages that float through your bloodstream like whispered requests. We need more blood cells, one message might say. The liver needs repair, another might whisper. New bone tissue is required here. And the stem cells respond with extraordinary generosity. They begin the remarkable process of transformation, choosing which type of cell to become based on what your body needs most urgently. This decision is irreversible. Once a stem cell commits to becoming a heart cell, it can never become anything else. But before that moment of commitment, stem cells exist in a state of beautiful uncertainty. They are like artists standing before a blank canvas, brush in hand, considering all the masterpieces they could create. They hold within themselves the potential for every type of cell that makes up your body. This potential comes from their unique relationship with their genetic books. While other cells have closed most of their genetic library and focus on reading just one section, stem cells keep their library fluid and open. They can access any chapter, read any page, follow any set of instructions written in their DNA. Scientists call this state pluripotency, but you can think of it more simply as unlimited dreaming. Stem cells are the dreamers of your body, imagining all the different lives they could live, all the different roles they could play in the symphony of your existence. Your bone marrow is not the only garden where these dreamers live. Small populations of stem cells exist throughout your body, tucked away in quiet corners like seeds scattered by a thoughtful gardener. Some live in your hair follicles, others in your intestines, still others in your brain. Each of these hidden gardens serves as a repair depot for its local neighborhood. When nearby cells become damaged or worn out, the local stem cells receive the call for help. They awaken from their patient waiting and begin the process of replacement and renewal. This is how your body has maintained itself throughout your entire life. The skin you had as a child has been completely replaced multiple times by new cells grown from stem cells. The lining of your intestines renews itself every few days, thanks to these cellular gardeners. Even parts of your brain benefit from fresh cells grown in hidden stem cell gardens. But as you age, these gardens become less active. The stem cells remain, but they respond more slowly to calls for help. They divide less frequently. They sometimes choose their new identities less precisely than they did when you were young. This natural slowing is part of the aging process, but it also represents an opportunity. What if scientists could awaken these sleeping gardens? What if they could convince aged stem cells to remember their youthful enthusiasm for transformation? Even more intriguingly, what if scientists could take your adult cells, the ones that have already chosen their life's work, and convince them to become stem cells again? What if a skin cell could be coaxed to forget that it is a skin cell and remember that it once held unlimited potential? This might sound like science fiction, but it is becoming science fact. Researchers have discovered ways to reset adult cells, to help them forget their specialized roles and return to their original state of unlimited possibility. These reset cells are called induced pluripotent stem cells, but you can think of them as cells that have learned to dream again. The process involves introducing specific chemical signals into adult cells, signals that say, remember who you were before you chose what to become. These signals activate genetic switches that have been turned off for years, sometimes decades. They awaken dormant sections of the genetic library that the cell had forgotten how to read. Slowly, gradually, the specialized cell begins to lose its commitment to its chosen identity. A skin cell stops thinking of itself as skin. A blood cell forgets how to carry oxygen. The cellular identity dissolves, and in its place emerges something remarkable, a cell that remembers how to become anything. These newly awakened stem cells can then be guided into becoming whatever type of cell is needed. With the right chemical encouragement, they can differentiate into heart cells, liver cells, brain cells, or any other type of tissue your body might require. As you breathe quietly in this peaceful moment, stem cells throughout your body continue their patient vigil. They wait in their hidden gardens, ready to serve, ready to transform, ready to become whatever your body needs them to be. But what happens when we take these patient gardeners and ask them to grow something entirely new? What occurs when we provide them with the perfect conditions to become not just individual cells, but entire organs? The answer lies in laboratories where scientists have learned to create artificial wombs made of glass and hope. In laboratories that hum softly through the night, something miraculous is taking shape. Behind glass walls, in chambers warm to the exact temperature of your body, new hearts are learning to beat for the very first time. These are not the hearts of unborn children. These are hearts grown from adult cells, coaxed back to their earliest dreams of what they could become. They pulse gently in their glass wombs, fed by artificial blood, breathing artificial air, yet somehow undeniably alive. This is where the journey from stem cell to organ begins. Imagine the most patient gardener in the world, tending to seeds that take months to sprout. This gardener knows exactly how much water each seed needs, precisely when to provide nutrients, exactly what temperature will encourage growth. They speak to their plants in the language of chemistry, offering gentle encouragement through carefully measured doses of growth factors and proteins. This is what scientists have become, gardeners of human tissue, learning to speak the ancient language that cells use to organize themselves into organs. The process begins with the stem cells we met in the previous chapter. These patient dreamers are placed into specially designed chambers that recreate the conditions they experienced when you were first forming in your mother's womb. The temperature must be perfect, the humidity exactly right, the chemical signals precisely balanced. But growing an organ is far more complex than growing individual cells. When you were developing before birth, your cells had to do something extraordinary. They had to learn not just what to become, but how to work together. Heart cells had to find other heart cells and link arms. They had to synchronize their beating. They had to form the chambers and valves that would pump blood for a lifetime. Scientists have discovered that cells remember these ancient choreographies. Given the right encouragement, stem cells will begin to dance the same steps that created your original organs. They cluster together in small groups. They begin to pulse in unison. They organize themselves into the complex three-dimensional structures that make organs work. The first sign that something magical is happening comes as a tiny flutter. In the glass chamber, a small cluster of cells begins to contract rhythmically. Beat. Rest. Beat. Rest. It is tentative at first, uncertain, like a baby bird trying to use its wings for the first time. But this rhythm is unmistakable. It is the signature of life itself, the heartbeat. The scientists who witness this moment never grow tired of it. After weeks of careful tending, after months of preparing the perfect conditions, they see this first fragile pulse and know they are watching the birth of a new organ. The growing heart is fed through tiny tubes that carry nutrients and oxygen, just as your umbilical cord once fed you. The glass chamber rocks gently, mimicking the movements that helped shape your organs before you were born. Mechanical pumps create subtle pressure changes that encourage the developing heart to grow stronger. Day by day, the heart becomes more sophisticated. Individual heart cells find their neighbors and form electrical connections. These connections allow the heartbeat to spread from cell to cell, creating the coordinated contractions that make a heart effective. The rhythm becomes steadier, stronger, more confident. Scientists can watch this development in real time using special cameras that peer through the glass walls. They see individual cells joining the growing network. They observe new blood vessels sprouting like tiny rivers through the developing tissue. They witness the formation of heart valves that will control the flow of blood. Each growing heart is unique, yet all follow the same ancient blueprint. The same genetic instructions that guided the formation of your own heart millions of heartbeats ago are now guiding these laboratory-grown organs. The cells remember their purpose across generations, across species, across the vast span of evolutionary time. But growing a heart that beats is only the first step. The real challenge is growing a heart that can integrate with a living body. The laboratory heart must learn to respond to the chemical signals that regulate heart rate. It must develop the electrical system that allows it to speed up during exercise and slow down during rest. This is where the collaboration between natural wisdom and human ingenuity becomes most apparent. Scientists provide the perfect environment, but the cells do the actual work of construction. Humans offer guidance, but the heart builds itself according to instructions written in DNA millions of years ago. The glass wombs that nurture these growing organs are marvels of engineering designed to mimic nature. They maintain precise temperature control. They provide exactly the right mixture of nutrients at exactly the right times. They create the mechanical forces that help shape developing tissue. Some chambers rock back and forth like a gentle cradle. Others create rhythmic pressure changes that simulate breathing. Still others provide electrical stimulation that encourages heart cells to beat in coordination. Each type of organ requires its own special environment, its own unique combination of chemical and physical signals. As the heart matures in its glass womb, it begins to resemble the organ beating in your chest right now. It develops the strong muscle walls that contract with each beat. It forms the smooth inner lining that allows blood to flow without clotting. It creates the electrical pathways that coordinate the rhythm of life. The scientists tend their growing organs with the devotion of parents caring for newborns. They monitor every heartbeat, adjust nutrients as needed, provide just the right amount of stimulation to encourage healthy growth. They are midwives to a new kind of birth, helping laboratory-grown organs come into the world. These glass wombs represent humanity's first tentative steps toward growing replacement organs from our own cells. The hearts beating softly in these chambers may one day beat inside human chests, giving new life to those whose own hearts have grown weary. But before these laboratory miracles can help real people, scientists must understand something even more fundamental. How do cells know exactly where they belong in the intricate architecture of an organ? The answer lies in the ancient blueprints that guide the construction of every complex structure in your body. Imagine the most magnificent cathedral you have ever seen. Picture its soaring arches, its intricate stonework, its perfect proportions that lift your eyes toward heaven. Now imagine that this cathedral built itself, stone by stone, with no architect to guide it and no blueprints to follow except those written in an ancient language that every stone could read. This is exactly what happens when your organs form. As you rest here peacefully, every organ in your body stands as testimony to the most sophisticated architecture in the known universe. Your heart is not simply a pump. It is a four-chambered marvel with precisely angled valves, electrical pathways that coordinate like a symphony, and muscle fibers arranged in spirals that create the most efficient pumping action possible. Your lungs are not merely bags for holding air. They are intricate branching trees with airways that divide and subdivide 37 times, creating a surface area the size of a tennis court folded into the space of your chest. Every branch, every tiny air sac, positioned exactly where it needs to be for maximum efficiency. Your kidneys are not simple filters. They are cities of microscopic tubes, each one precisely engineered to clean your blood while preserving everything your body needs to keep. A million tiny factories working in perfect coordination, creating a filtration system more sophisticated than any human engineer has ever designed. How do cells know how to build such complex structures? How do they coordinate their efforts across millions of individual decisions to create organs that work with such precision? The answer lies in something scientists call morphogenesis, but you can think of it more simply as the art of cellular architecture. Your cells are master builders who carry within themselves not only the materials for construction, but also the complete blueprints for every structure they might ever need to build. These blueprints are written in the same four-letter genetic language we have explored throughout our journey. But unlike the simple instructions that tell a cell how to make a protein or perform a basic function, architectural instructions are incredibly complex. They must specify not just what to build, but where to build it, when to build it, and how to coordinate with millions of other builders working on the same project. The construction of an organ begins with signals. Chemical messages that float through the developing tissue like whispered instructions passed from worker to worker on a vast construction site. These signals tell cells where they are in the growing structure. They provide coordinates in three-dimensional space. They say, "You are here," in this part of what will become the heart, or your position is there, in what will become the left lung. Cells listen to these positional signals and respond by reading the appropriate section of their genetic blueprints. A cell that receives the signal you are in the heart begins reading the heart construction manual. A cell that hears you are in the kidney opens the kidney architecture guide. But the true magic happens when cells begin to communicate with their neighbors. As they read their construction instructions, they start sending out their own chemical signals. These signals tell nearby cells what type of tissue is being built in their area. They coordinate the work so that blood vessels grow where blood vessels are needed, and nerve connections form where nerve connections belong. This communication creates what scientists call pattern formation. It is like having thousands of builders on a construction site, each one working on their own small section, yet somehow coordinating their efforts to create a unified structure. The heart valve builders must align their work with the heart chamber builders. The kidney filter builders must connect properly with the kidney tube builders. The cells accomplish this through an ancient system of chemical gradients. Imagine a construction site where different types of paint are slowly spreading through the air. Workers can smell the paint and know what type of work is being done in different areas. They can follow the scent gradients to find where their own type of work is needed. Cells use molecular gradients in much the same way. Special signaling molecules spread through developing tissue, creating invisible maps that cells can read and follow. A cell can sense how close it is to a blood vessel by detecting the gradient of blood vessel signals. It can determine its position in a growing organ by reading the combination of different molecular signals that surround it. These gradients change over time as the organ develops. What begins as a simple pattern becomes increasingly complex as new types of cells join the construction project. The molecular landscape shifts and evolves, providing updated instructions to cells as the organ takes shape. Perhaps the most remarkable aspect of cellular architecture is how cells know when to stop building. A heart must be exactly the right size for the body it will serve. Lungs must have precisely the right number of air sacs. Kidneys must contain exactly the right ratio of filtering units to supporting tissue. Cells accomplish this through feedback systems that monitor the progress of construction. As an organ approaches its proper size, the cells begin producing different signals. These signals gradually reduce the rate of new construction and begin the process of fine-tuning and refinement. The final stages of organ development involve a process called differentiation refinement. Cells that have been working as general construction workers begin specializing in their final roles. Heart muscle cells develop the proteins needed for strong, coordinated contractions. Lung cells create the delicate surfaces needed for gas exchange. Kidney cells perfect their filtration mechanisms. Throughout this entire process, cells are constantly checking their work against the genetic blueprints. If mistakes are detected, repair mechanisms spring into action. Cells that have developed incorrectly may receive signals telling them to try again, or to undergo programmed death to make room for properly formed replacements. The result of this extraordinary construction process is the collection of organs that keep you alive at this very moment. Each one a masterpiece of biological architecture, built by cells following instructions that have been refined over millions of years of evolution. But understanding how organs normally develop is only the first step toward growing replacement organs in the laboratory. Scientists must also learn how to recreate the complex symphony of signals and gradients that guide cellular construction. This knowledge becomes especially important when we consider the ultimate goal of regenerative medicine, growing organs that can help heal the children and adults who need them most. Soon, we will explore how this ancient wisdom of cellular architecture might offer hope to those whose own organs have forgotten how to work properly. Tonight, somewhere in the world, a 7-year-old girl is sleeping in a hospital bed. Her name is Emma, and her heart is too tired to beat properly. She has been waiting for 2 years for a new heart, hoping that somewhere, someone her size will become an organ donor. But Emma's story is not unique. In children's hospitals around the globe, thousands of young patients wait for organs that may never come. They need new hearts, new kidneys, new livers. They need the gift of healthy tissue to replace parts of their bodies that never formed correctly, or that stopped working too soon. These children carry within their small bodies the same remarkable stem cells we have explored throughout our journey. The same cellular architects that built their organs before they were born. The same molecular libraries containing the blueprints for every tissue they could ever need. The tragedy is not that their bodies lack the instructions for health. The instructions are all there, written in the four-letter alphabet of their DNA. The tragedy is that sometimes, in the complex process of building a human being, mistakes happen. Sometimes the architectural signals get crossed. Sometimes the cellular construction crews receive the wrong instructions. A heart develops with chambers that don't connect properly. Kidneys form without the right number of filtering units. A liver grows with passages that lead nowhere. These are not failures of design. They are simply the inevitable imperfections that occur when something as complex as a human body builds itself from a single cell. For most of human history, children born with these conditions had few options. Doctors could repair what they could, support what they couldn't fix, and hope for the best. Organ transplantation offered hope for some, but finding matching organs for children is especially difficult. Adult organs are too large. Pediatric donors are tragically rare. But now, in laboratories where scientists work with the patients of gardeners, a new kind of hope is growing. The same technologies we have explored in our journey, CRISPR and stem cells and cellular architecture, are coming together to offer something that seemed impossible just a few years ago, the possibility of growing replacement organs from a child's own cells. Imagine taking a small sample of skin cells from Emma, the 7-year-old with the tired heart. These skin cells carry within them the complete genetic library needed to build any organ in her body, including a healthy heart. The challenge is convincing these adult skin cells to remember their earliest dreams of what they could become. Scientists have learned to speak the chemical language that can reset these cells, helping them forget that they committed to being skin and remember their original potential. The process is like a gentle conversation, using molecular signals to whisper, "You don't have to be skin forever. You can dream again." Once Emma's cells remember how to dream, they can be guided into becoming heart cells. Not just any heart cells, but heart cells that carry Emma's exact genetic signature. Heart cells that her immune system will recognize as belonging to her, not to a stranger. These heart cells can then be placed in the glass wombs we explored earlier, where they will receive the chemical signals and mechanical encouragement needed to organize themselves into a functioning heart. A heart grown from Emma's own cells, sized perfectly for her 7-year-old body, beating with the rhythm that will carry her through childhood and beyond. This is not science fiction. This is science in progress. Around the world, researchers are working to make this vision a reality. They are refining the techniques for cellular reprogramming. They are perfecting the conditions needed to grow organs in laboratory settings. They are learning to recreate the complex signals that guide organ development. The first successes have been with simpler tissues. Scientists have grown patches of heart muscle that beat in laboratory dishes. They have created sections of blood vessels that pulse with life. They have coaxed cells to form the basic structures of kidneys, lungs, and livers. Each success brings us closer to the day when children like Emma will have access to organs grown from their own cells. Organs that will never be rejected by their immune systems. Organs that will grow with them as they mature from children into adults. But the implications extend far beyond individual healing. The same techniques that could grow organs for children with birth defects could also help adults whose organs have been damaged by disease or injury. The same cellular reprogramming that could save Emma's life might one day help millions of people around the world. The beauty of this approach is that it works with the body's own wisdom rather than against it. Instead of forcing the immune system to accept foreign tissue, we help the body heal itself using its own cellular resources. Instead of fighting the natural processes of organ development, we recreate the conditions that made those processes work in the first place. There are still challenges to overcome. Growing complex organs remains difficult. Ensuring that laboratory-grown organs integrate properly with existing blood vessels and nerve connections requires more research. Making these treatments safe and widely available will take time. But the foundation has been laid. The scientific principles are understood. The basic techniques are being refined every day in laboratories around the world. Children who are born next year may live in a world where organ failure is no longer a life sentence, but simply another medical condition that can be treated with organs grown from their own cells. As Emma sleeps in her hospital bed tonight, scientists in laboratories continue their patient work. They tend to growing tissues with the devotion of gardeners nurturing precious seedlings. They refine techniques with the precision of craftsmen perfecting their art. And perhaps most importantly, they carry in their hearts the same hope that Emma's parents hold, that science and compassion, working together, can create miracles that seemed impossible just a generation ago. The future they are building is one where every child born with organ problems carries within their own body the seeds of their own healing. Here, in the gentle quiet of this moment, as your breathing slows and your muscles relax, you are connected to one of the most extraordinary stories ever told. It is not a story from a distant future or a far-off laboratory. It is the story unfolding right now, inside your own body, in the trillions of cells that make up the miracle of you. Every cell in your fingertips carries the same potential that scientists are learning to harness in their glass wombs and cellular gardens. Every cell in your heart holds the same ancient wisdom that guides the growth of laboratory organs. Every cell in your body is both a participant in your current life and a keeper of possibilities for healing you cannot yet imagine. As you rest here tonight, your cells are writing the future of medicine with their quiet, persistent work. In your bone marrow, stem cells are dividing slowly and purposefully, creating fresh blood cells that will carry oxygen through your body tomorrow. These same stem cells carry within them the potential to become heart cells, liver cells, kidney cells, or any other type of tissue you might ever need. They wait patiently in their hidden gardens, ready to serve, ready to heal, ready to become whatever your body asks of them. In laboratories around the world, scientists are learning to speak the language that these cells already know. They are discovering how to ask stem cells to remember their unlimited potential. They are perfecting the art of cellular conversation, using chemical signals to guide cells toward becoming the organs that sick people need. But the most remarkable part of this story is that you do not need to wait for the future to benefit from your body's healing wisdom. Right now, as you breathe slowly and peacefully, your cells are using these same principles to maintain and repair your body. Your liver cells are dividing to replace old, worn-out tissue. Your skin cells are constantly renewing the protective barrier that keeps you safe from the outside world. Your intestinal cells are replacing themselves every few days, ensuring that your digestive system remains healthy and efficient. All of this happens without your conscious thought, guided by the same cellular intelligence that scientists are learning to direct in laboratories. The cuts on your skin heal because your cells remember how to rebuild damaged tissue. Broken bones knit back together because bone cells retain the architectural knowledge needed to restore proper structure. Your immune system fights off infections because immune cells carry within them the ancient wisdom of recognizing friend from foe. You are not just a static collection of organs and tissues. You are a dynamic, self-healing system that is constantly rebuilding itself from the inside out. Every 7 years, most of the cells in your body will have been replaced by new ones. The you that exists today is built from different cellular materials than the you that existed 7 years ago, yet you remain unmistakably you. This constant renewal is possible because your cells carry within them not just the instructions for what they currently are, but also the memory of what they came from and the potential for what they could become again. Your skin cells remember that they descended from stem cells. Your heart cells retain the genetic pathways that could, under the right conditions, help them repair significant damage. The CRISPR technology we have explored in our journey works because it harnesses the editing systems that your cells already use to maintain their genetic libraries. The precision molecular scissors that scientists employ in laboratories are refined versions of tools that bacteria developed billions of years ago, and that exist in modified forms in your own cells today. The organ-growing techniques being perfected in research centers around the world succeed because they recreate the same conditions and signals that guided the formation of your own organs before you were born. The glass wombs and chemical gardens of regenerative medicine are human attempts to duplicate the nurturing environment that your mother's body provided when you were developing. As you settle deeper into rest, consider this profound truth: you are already living proof that the most ambitious dreams of regenerative medicine are possible. Your body grew itself from a single cell. Your organs formed themselves according to genetic blueprints. Your tissues learn to work together in the symphony of your existence. The future of medicine is not about inventing entirely new processes. It is about understanding and directing the processes that already exist within you. It is about learning to speak fluently the language that your cells have been using all along. In the years to come, children born with heart defects may receive new hearts grown from their own cells, hearts that will grow with them as they mature. Adults whose organs have been damaged by disease or injury may receive replacements that their immune systems will welcome as their own. The elderly may have access to tissues that help them maintain their health and vitality far longer than previous generations. But all of these future miracles will be based on the same principles that govern your body right now. The same stem cell potential. The same cellular communication systems. The same architectural wisdom that built you in the first place. You are not separate from the future of medicine. You are carrying it within you at this very moment. Every breath you take is supported by cells that contain the blueprints for healing. Every heartbeat is powered by tissue that remembers how to rebuild itself when damaged. As you drift towards sleep, your cells continue their quiet work of maintenance and renewal. They replace damaged proteins. They repair tiny breaks in DNA. They clear away cellular debris and prepare your body for tomorrow's activities. They do all of this while you rest, using the same molecular tools and genetic wisdom that scientists are learning to harness for growing organs and healing disease. The future of regenerative medicine is not coming from outside your body. It is emerging from within you, written in the four-letter alphabet of your DNA, carried in the patient dreams of your stem cells, expressed in the endless creativity of your cellular symphony. Tonight, as sleep embraces you, you can rest in the knowledge that you are both the inspiration and the source of tomorrow's healing miracles. The future is growing within you, one cell at a time, as naturally as your breath and as quietly as your dreams.